Good morning, children. In the last class, we have completed until then the topic artificial hybridization, right? So we have discussed about the number of things. It is nothing types of pollination, then after the pollen-pistil interaction, and also we have discussed about the artificial hybridization. Today in this class, we will continue the topic the double fertilization. See, observe the diagram, whatever they have given neatly. In the first diagram, the left side, they have given the complete structure, and that is the indication of the pollination. The pollen rise is placed on the surface of the stigma. Then after, you can identify in the second diagram, the growth of the pollen tube, isn't it? Inside the pollen tube, there are two sperms, that is nothing but male gametes, moving towards the ovary. And finally, in the third diagram, in the enlarged structure, you can identify that is penetration. That's what we have discussed in earlier class, the pollen-pistil interaction, isn't it? Yes. Now let us discuss the what is meaning of the double fertilization. Yes. See, after the entering of one of the male gamete, that is one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergids. So out of these two, one male gamete moves towards the egg cell and fuses with its nucleus. So this process is also known as syngamy, to form the zygote, that is diploid, because male gamete already containing N and the egg containing N. By the fusion of these two nucleus, it will be give stage to the diploid zygote. Then what about the other one? The other male gamete moves towards the two polar nuclei located in the central cell and fuses with the, that means the polar nuclei. You can identify and observe. Yes. What happens now? When the other male gamete move towards the polar nuclei located in the central cell and fuses with them to produce triploid, that is already polar nuclei containing 2N and the male gamete is N. By the fusion of these two, the triploid primary endosperm nucleus, as it involves the fusion of 3 haploid nuclei, that means totally 3N. It is called triple fusion. Remember the double fertilization and triple fusion. Got it? So then only it will be considered as the fertilized embryo sac. Got it? Yes. So after this one, what will happen? The process, as it continues, since two type of the fusion, that means syngamy and triple fusion takes place in an embryo sac, it is called double fertilization and triple fusion. It is an event unique to the flowering plants. Remember, so this type of the double fertilization cannot be seen in the earlier. We cannot see this type of the characteristic features in, that is, gymnosperms or pteridophyte, bryophytes. These are all, they will not possess this type of the character. That means double fertilization and triple fusion is the unique character, that is one and only. Unique means one and only, which cannot be identified in any other groups. So that is the double fertilization is the unique characteristic features of the flowering plants. Got it? Yeah. And now the process will continue in the central cell after the triple fusion. What will happen? After the fusion of this central cell, that means triple fusion becomes PEC or PEN also we can say, that is primary endosperm cell, primary endosperm nucleus. Okay, and it develops into the endosperm. Endosperm means it is a nutritive tissue. Okay. And the zygote, what will happen? The fate of the zygote. The zygote further develops into an embryo. Okay. Yeah. See, after the the fertilization process, what are the structures and the events we can identify? So those will be considered as the post-fertilization structures, and we can also say the post-fertilization events. Majorly, there are 4 post-fertilization events can be seen in angiosperms, because earlier class we have discussed in detail the pre-fertilization structures and events and the fertilization structures and events, right? Yeah. Then after that, post, that means after fertilization. Got it? Yeah. Here is the briefly they have given in the diagrams. Are you observing? Observe briefly. Later we will discuss one by one. Okay? Yeah. So that means the first fertilization events. Majorly there are 4 events we can identify in angiosperms. The formation of endosperm, which is also known as endosperm development. Then the development of embryo, that is embryo development. And we can also identify the ovules. It develops into form seeds. Okay. Remember, in all the cases we cannot expect the seeds. At the same time, some species, the ovary develops into the fruits. Okay. So that means both the structures also it can be identified in some species, but in some species the ovaries develops into seeds, and later ovary develops into a fruit. Got it? Here's a brief diagram. In that one, first one you can identify, yes, egg cell, and it inside the embryo sac containing polar nuclei. It is indicated in the yellow dots, the red dot indication of the egg, and when one male gamete in the second diagram you can identify fuses with the sperms, and it will be gives rise to the zygote, while the other male gamete fuses with the, that is primary endosperm nucleus. Got it? Yeah, that is polar nuclei fuses with the sperms, gives rise to the endosperm, that is primary endosperm nucleus. And as I said now only, yeah, the formation of the ovules into seeds and ovary into fruits. Also, you just observe here, again we will discuss the zygote, how it will develop. In the beginning stage, one cell and 2 cell, later globular embryo, then it will be get converted into heart shape, then later the mature embryo. Got it? Yeah. So these are all the events. Let us discuss one by one. And that one, first one is, yeah, formation of endosperm, or we can also say endosperm development. For the purpose of understanding, in the middle we have given that additional diagram, but in the left side the textbook diagram. You can observe this one, both one. Okay, so that you can easily understand that means formation of endosperm topic. So after the fertilized embryo sac, what will happen? What are the events? See, as here it is given, the primary endosperm cells start dividing, that means divides repeatedly to form a replied endosperm tissue. Yes, then so these endosperm cells are filled with the other food materials. They are used for nourishment, that is nutrition of the developing embryo. So observe neatly what are the things happens here. The first one, in the diagram only you can identify the degeneration means disappearing of the synergies, and the zygote starts development. Got it? Now primary endosperm cell and the formation of primary endosperm nucleus, degenerating adipodal cells. It is disappearing. Got it? I hope you are observing keenly. Got it? Yeah, fine. And here you can identify How it will be? The step-by-step mechanism. Generally, in common endosperm development, that is PEN, primary endosperm nucleus, which is undergoes successive nuclear divisions. That means repeatedly to give rise to the free nuclei. That is, it is not in the beginning stage. It is not, found on the surface. It is floating in the liquid. Then it will be considered as a free nuclei. This stage, it is also called free nuclear endosperm formation. Okay. Still, it is not yet settled on the wall of the inside. Okay. Then it will be considered as a free nuclear endosperm. And later, the number of free nuclei varies greatly from species to species and also from family to family, because there are a number of families and also species they are found in angiosperms, right? Yes. So like that, the number of free nuclei, it is differs greatly from one species to another species. Got it? Then endosperm becomes cellular due to the cell wall formation, and as the deposition increases, the thickness of the wall also increases. See, here is the best example. Free nuclear endosperm, also cellular endosperm, which can be identified in the coconut. In case of the tender coconut water, it is a very best example. The free nuclear endosperm is made up of thousands of nuclei, and the surrounding white kernel or copra, generally is white kernel or copra is the cellular endosperm. Then you can ask me the question. Sometimes the question arises. Sometimes the tender coconut is not sweet. That means still free nuclear endosperm is not formed. Okay, I hope you are experienced. I think you must have experienced. If the tender coconut water is sweet, it is containing thousands of free nuclear endosperm. Okay, but inside that one, if that tender coconut containing very thick white kernel, that means cellular endosperm is present. Okay, so you must have experienced these things also. Got it? So best example. Sometimes in competitive also they will ask. So this is the only species where we can identify both free nuclear endosperm and cellular endosperm clearly. Got it? Yeah. So, and how this embryo development? As I said, in the beginning, the endosperm is very important. Without the endosperm, that means there is embryo development will not take place, because for the purpose of nourishment of the embryo, endosperm is required. Just think about it. If there will be no nourishment, how the embryo develops? Isn't it? That's why the first stage is the formation of endosperm. Then later embryo development will start. Got it? Here is briefly, as I said, the zygote. Then it will be turned into the pro-embryo stage. Then later it will develop into the globular stage. You can identify the diagrams. And then it will turn into the heart-shaped diagram. Yes, finally mature embryo. Okay, so the embryo develops at micropylar end of the embryo sac, where the zygote is situated, isn't it? In the earlier classes only we have discussed, because where the egg generally it is present toward the micropylar end, right? Yes, there only that embryo developments will be takes place. Got it? I hope you can easily identify the structure. Most zygotes divide only after the formation of some endosperm. So this gives nutrition to developing embryo. Okay, isn't it? As I said, so if there will be no endosperm, there will be no nourishment. Got it? Then, based on the type of the cotyledons in monocots and dicots, so it differs greatly. But embryogeny, it is quite natural and normal. In the early embryonic developments, it is similar. Only after the complete clear structure formation, then only it will be differs with respect to the cotyledon. In monocots, it will become monocotyledon, and in dicot, it will become a two cotyledon structure. Got it? Yes. If we take an example, let us discuss here a typical dicot embryo and the typical monocot embryo structures. It is given. It is present in your textbook also. You can just check it out. Okay. The structure, what it will be containing. If you just observe the first dicot embryo only, the plumule, the cotyledons, then that is two cotyledons, hypo, yes, below cotyledon, and the radicle, it is surrounded by root cap, isn't it? You know very well that the last year only we have discussed. The plumule is nothing but youngest shoot system, isn't it? Yeah, radical is youngest root system. Later it will be developed into the root system, and the plumule later it will be used to the shoot system or stem. Yeah. At the same time, you just observe here is also, if you see the LS of the grass, that is embryo, the best example of the monocot. It is present in our textbook, isn't it? Yeah. It has an embryonal axis and two cotyledons. In the dicot structure, two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyle, which terminates with the plumules. That means ends. In generally, we can say a stem tip also. The cylindrical portion below the level of the cotyledons is hypocotyle, that terminates with the radicle. That is nothing but root tip. The root tip is very delicate structure, so that's why it is covered and protected with a root cap. Got it? Yeah. So this is about the dicot. Then when you take an example of the monocotyledon embryo, yeah, just observe the differences along with the structure. They possess, that means monocotyledon embryo, it possesses only one cotyledon. The meaning itself, it is present. It containing single cotyledon, right? Yeah, that's right. Then the cotyledons of the grass family. In generally, I told you number of times, the grass family is commonly known as Graminae or Poaceae. Yes, the cotyledons of the grass family is called scutellum, and it is situated lateral to the embryonal axis. At its lower end, the embryonal axis has a radicle and root cap enclosed in coryza. Yeah, remember, the coryza is an undifferentiated sheath. Sometimes 1 mark question also they will ask, okay, where the coryza is present, or the radicle is surrounded by, like that they will ask the question. And if you observe the structure, the portion of embryonal axis above the level of attachment of the scutellum is the epi. Epi means above, upper region, that is epicotyle, and it has a shoot apex and a few leaf primordia. Very youngest structure. Leaf primordia is nothing but very youngest these structures enclosed in a polyptile, a hollow of foliar structures. Got it? Yeah. Later it will be developed into the, that means, yeah, leaf. That means complete leaf structures. So this is about the cotyledons. Got it? Yeah, 4 marks questions are sent and 5 marks questions also they will ask the diagram along with the explanation. Okay. So this is about the monocotyledonous embryo. Yeah. So let it be. So next class we will continue the remaining thing. Thank you.